Thank you very much. Ladies and gentlemen, dear colleagues, dear students, after this fascinating talk about the elephant sensory system, I'm going to give you an introduction about the human sensory system and specifically related to pain. And I changed my title a little bit to be a little broader, and I'm trying to give you some ideas how we can develop new drugs. At the Department of Pharmacology and Pharmacotherapy at the University of Pécs, we have a long history of sensory research and pain research. We do research, but also some drug development and innovation projects. Just before I start with some specific information, I'm trying to show you very briefly the pain sensing and pain processing pathways. Of course, the story starts at the periphery. We have the peripheral nociceptors, which are specific structures responding to different stimuli. And then these peripheral nerve endings, the cell bodies of which are located in the dorsal root ganglia or in the facial area, in the trigeminal ganglia. And the central projection, the central terminals, they go to the spinal cord, the spinal dorsal horn. They switch to the secondary sensory neurons, and then the pain signal is going up into the brain. Of course, it's a very complicated processing in the brain, in the periaqueductal gray, and also in different brain regions. But then finally, we have the pain sensation. Pain is a human terminology, so we cannot evaluate pain in animals. We can only evaluate the pain defensing behavior, which is called nocifensive behavior, but pain is a subjective sensory emotional experience which is associated with real or potential tissue damage. And this is the latest definition of the International Association of the Study of Pain. So pain is subjective and unpleasant. Nociception can be measured in animals. So this is what indicates the nociceptor activation. And we have some terms that I would like to determine at the beginning to understand later what I will talk about. Hyperalgesia is a pain sensation to mild painful stimuli. So when this increases, but basically the stimulus is painful, mildly painful. Allodynia is when we have pain sensation to absolutely non-painful stimuli, like just mild warm water. And then hypoalgesia or analgesia, that's reduced or absent pain sensation, and analgesic, that means painkiller. Okay? So why is it important to study pain? Because the chronic, long-lasting pain is a real unmet medical need. Pain is basically a good thing, because if we have an acute pain, we can actually defend ourselves from different injuries. But when we have chronic, long-lasting pain, that is actually reducing the life quality of patients and their work quality, and this is very, very debilitating. And actually, the chronic, long-lasting pain is very, very often so-called neuropathic. Neuropathic means that it is caused by a lesion or disease of the somatosensory nervous system due to different trauma, nerve injury, accident, for example, or operation, inflammation, like in long-lasting rheumatoid arthritis or other joint inflammation, or metabolic reasons or toxicity, and the prevalence is quite high. And the main symptoms are, of course, the loss of function of the somatosensory system or abnormal or increased pain sensation like paresthesia, dysesthesia, hyperalgesia, allodynia. These two are actually just strange sensations. And actually, we don't know what the reason is. We don't have real pathophysiological mechanisms in the background that we know. So that's for sure that the role of the neuro, the sensory, the immune and the vascular system as a complexity, it is very important in the pathophysiological pathways. So it is important to understand what's going on because the therapy is still an unresolved issue. So why do we need novel analgesics with novel mechanisms of action? The answer is very simply because in the last many centuries, we haven't had any new drugs on the market. We actually did new drugs, but with the same mechanism, with the already existing, basically two mechanisms. Actually, we still only have the non-steroidal anti-inflammatory drugs, which inhibit the cyclooxygenase enzyme and inhibit the production of prostaglandins, like aspirin, diclofenac, ibuprofen, paracetamol. Aspirin we have already heard about in Professor Ciechanover's talk. So that's a very old story, and they do not act in chronic neuropathic pain. And the other group is opioids. This is also a very, very old group of drugs like morphine, codeine, fentanyl. They are strong analgesics, but in very many percentage, they are also ineffective. And also, after long-term use, they have many, many side effects related to the gastrointestinal system, the kidney. They cause obstipation, respiratory depression, and so on. And we have a third group of analgesics besides these two very well-known, very old group of drugs, and those are the adjuvant analgesics, certain anti-epileptics and antidepressants, but they also have a lot of problems, and they are also not always effective. So why hasn't been the analgesic development so successful in the last centuries? Because the research did not really focus on the patient's heterogeneity. There were very few studies starting from the clinics. There were no well-designed clinical trials. And on the other hand, we didn't have, and we still don't really have, perfect animal models with translational relevance. And these models did not really focus on the complexity of the problem. And then we also had no really appropriate investigational techniques, complex investigational techniques. So what happened in the last few decades? There have been some solutions and improvements. It is important to select the patient cohorts, have registries, to have broad patients registries, to have more translational chronic animal models, and high sensitive analytical techniques like functional magnetic resonance imaging of the brain. And we should have hypothesis-free kind of omics approach, and this developed a lot, like determining the mRNA transcripts, proteomics, the proteins, and the metabolites in the human cohorts. And also we have now big data analysis and bioinformatics tools. And then in the next few minutes, I'm showing you 3 directions that we have been performing in the last years and some relatively successful data that we obtained. Our research group focuses on the primary sensory neurons and a subpopulation of the primary sensory neurons, which are called capsaicin sensitive primary sensory neurons, which actually create 2/3 of all sensory neurons. And the cell bodies are located in dorsal root ganglia. The peripheral terminals go to the periphery, to the skin, the joints, internal organs, and the central projections go to the spinal dorsal horn. And why are these capsaicin sensitive? Very important and unique. They are called capsaicin sensitive because they express the receptor for capsaicin in the membrane, and they are unique because besides sensing pain and actually giving information to the brain about painful stimuli, all sensory neurons can do that. They also release mediators, and these mediators are also inflammatory and anti-inflammatory. The inflammatory mediators induce so-called neurogenic, because it's neural dependent inflammation, edema, vasodilation, inflammatory cell activation in the periphery, and they also release anti-inflammatory mediators like a peptide I'm going to talk about called somatostatin, and this is getting into the systemic circulation and exerts inhibitory, anti-inflammatory and analgesic actions throughout the body. So for these unique triple functions, these capsaicin sensitive subpopulation of sensory nerves are really important. And our team focuses on what mediators are released, how these nerve endings modulate the inflammatory processes, both in the periphery and in the central nervous system, and how these nerve endings are activated. They are activated by a very broad range of stimuli, like prostaglandins, protons, and other mediators I'm going to talk about. And there are some receptors called the transient receptor potential receptors that I'm also going to talk about. So we focus on all these mechanisms, and based on this, there are 3 main targets that we can target for developing or identifying novel analgesics. One is to inhibit the inflammatory process, both at the periphery and in the central nervous system, by inhibiting the different inflammatory mediators. Then there is another option to actually activate the inhibitory mechanisms that we have already identified and somehow try to activate the receptors, like, for example, one receptor for this inhibitory peptide. And the third mechanism is to inhibit the activation of these nerve endings. Okay? And I'm just giving you some impressions of all these three. So the first is how we can inhibit the inflammatory processes at the periphery and in the CNS. And for this, we have a project which led to a so-called drug repurposing. In the last years, repurposing means using a drug that is already existing on the market for a different indication. And that's quicker and easier because the drug is already registered. And for this, we selected a disease which is a kind of neuropathic pain. It is called complex regional pain syndrome, which occurs after a small injury, which is normally healed. But in certain patients, this leads to extreme constant pain with hypersensitivity, edema formation. This is how it looks like for years. It just doesn't heal. And we don't know what this is about, what the reason is, what the mechanisms are, but this is for sure that complex neuroimmune interactions and autoimmunity are suggested to be involved in these pathophysiological processes, and actually the therapy is very, very unresolved. Nothing is really effective. And actually there is no good animal model, because if we don't know what the mechanism is, of course we can't create a good animal model. And a very good example for this is a collaboration of my group with a group in Liverpool, Walton Pain Center. They are clinicians and they have complex regional pain syndrome patients, and they treat these patients by plasmapheresis, which means that the immunoglobulins are removed from the plasma together with a lot of other inflammatory mediators. And the idea was that this disease has a kind of autoimmune mechanism. So they identified immunoglobulins, IgGs, immunoglobulin Gs, and we injected these human immunoglobulins to mice, and we did a small incision on the mouse paw that mimicked the small injury that happens in animals. And then after doing this small injury, which is normally healed in the mice, we treated one group of mice with healthy human immunoglobulin and the other group of mice with diseased patients' purified immunoglobulins. And then we measured the main symptoms of complex regional pain syndrome in animal models. And this is a summary. We published that in 2014. Actually, not all the symptoms of the disease could be mimicked, but the most important symptoms, which are pain, this is mechanical hyperalgesia, touch sensitivity increase. This is what we can measure in animals. And the edema and some inflammatory mediator increase could be mimicked in this animal model. So that was a successful animal model. We had 2 papers, and we proved that the main symptoms of the disease can be transferred by the patient's immunoglobulin. So actually autoimmunity is confirmed in the pathophysiology of the disease. And we also proved in this model that it has nothing to do with the peripheral inflammation. All the inflammatory parameters are unchanged, but there is a change in the central nervous system, and there is a neuroinflammation in the central nervous system. And also we proved that these neuroinflammatory mechanisms are related to the central sensitization to pain at the level of the spinal cord and the brain. And there is a key mediator, an inflammatory cytokine you might have heard of. It's a very well-known cytokine called interleukin-1, IL-1. And this is involved in the central nervous system in the pathophysiological process. And then we did, and this is a new approach, we did a transcriptomics analysis. So we measured a broad range of messenger RNAs with the deep sequencing of the dorsal root ganglia, of the primary sensory neurons. And we found a lot of genes. It's not important what they are, but we found a lot of genes. It's clearly differentiated. This is the CRPS, complex regional pain syndrome mice, and these are the healthy ones. Several genes are upregulated and several are downregulated. And we did a bioinformatics analysis, and I just highlight here that the cytokines and the cytokine signaling and neuroinflammation came out also from this analysis. And we identified here the interleukin-1 role, and also TNF, that's another inflammatory cytokine, small glycoprotein signaling inflammation, and another signaling pathway called the JAK-STAT pathway. And here I summarized what they are. So interleukin-1 signals through a signaling pathway inflammation and immune responses in the neurons, which leads to inflammation and pain, and also TNF, and also this JAK-STAT, that's the Janus kinase, and the STAT, this signal transducer activator of the proteins, they turned out to be important on the basis of this unbiased analysis. And fortunately, we have already drugs targeting this, although not for pain, but for other autoimmune diseases like rheumatoid arthritis, psoriasis, and anakinra, that's an IL-1 inhibitor, etanercept is a TNF inhibitor, and this tofacitinib, that's a JAK inhibitor. And they are used for other reasons, but they are already in the market. And we tested the effect of these drugs in the animal models. And I just show you here. Here I showed the mechanical hyperalgesia, so the pain threshold decrease. So the lower this, the more pain the animal has. Because we measure this with this blunt end needle, we touch the paw, and the animal withdraws the paw, and that's the threshold. And if this decreases, we have more pain. So here the red line shows that the animals have pain if they are treated with the diseased patient's immunoglobulin, and if we treat the patients with anakinra, the pain is disappeared. It's just like the normal. And also, if we start the treatment late, if we don't start the treatment at the very beginning, we measure these mice for a long time, but we just start the treatment later, and this is what mimics the clinical condition, it is still effective. So that's quite promising. And also, etanercept and tofacitinib, the two other drugs, the TNF targeting and the JAK-STAT signaling drug, they also reduce, actually abolish, this pain response in the mice, which are induced by the diseased IgG, immunoglobulin. So as a conclusion of this first part is that the main symptoms can be mimicked in our animal models, so it can be transferred by the diseased patient's immunoglobulin. So we could prove the role of autoimmunity, and we also proved that neuroinflammation and central sensitization are related to neuroinflammatory mechanisms. We identified some main targets, and we could prove that these drugs like anakinra, etanercept, and tofacitinib, they are effective in our animal models. So they might be useful in the clinical practice for drug repurposing or drug repositioning. And based on these data, and this is quite good news, a small clinical trial started in Liverpool at our collaborator site in January, and we are looking forward to the data to have clinical proof for this. Okay, for the second part, and I will be shorter here, this is original drug research and original drug development because we identified an inhibitory peptide, somatostatin, released from these nerve endings, activating a specific receptor of somatostatin, somatostatin 4 receptor. That's a peptide. We are searching for agonists, so activators of this receptor, whether they can be useful for pain decrease. Somatostatin is a peptide which has a lot of different functions because it has 5 different target molecules, but we are not really interested in any others, just this somatostatin 4 receptor, because in the last 15 years we provided a broad range of evidence that this is responsible for the anti-inflammatory and the analgesic effects. So actually we focused on this one. And then we searched for the expression of these receptors in different brain regions. I'm not showing you all the data, but in the somatosensory cortex, you can see the red dots, and the red dots represent the mRNA for the somatostatin 4 receptor. So we could prove that this is present in the somatosensory cortex and several other pain percepting brain regions, like the hippocampus in the brain and the habenula. These are all the red dots representing the mRNA for this receptor. And we also characterize the neurons, and they are excitatory neurons. And then we have some small compounds together with chemists which activate this receptor, and we published several papers that these compounds, which could be patented, could be promising drug candidates. And this is a model in animals. It's a purely neuropathic pain model when we ligate the sciatic nerve, 1/3 of the sciatic nerve, and this nerve injury induces hyperalgesia, so drop of the pain threshold. We also use this touch sensitivity, and these 4 compounds here, you see the anti-hyperalgesic or analgesic effect. All of them inhibit by about 50–60% in microgram per kilogram dose the hyperalgesia, the pain response, which is a good thing because actually none of the reference compounds can do that. So now we actually proved that in neuropathic pain and also in acute stress and chronic stress, which I didn't show you because of the lack of time, this receptor is an interesting target. If we activate this receptor, it can lead to analgesic and anti-inflammatory actions, but also, meanwhile, anxiolytic and antidepressant actions, which is good because chronic pain is very commonly accompanied by chronic stress. And now we have the drug target identified. We have some small molecules which are patented, and this is at this stage at a very early just drug discovery and very early drug development phase, and now we are working on the lead molecule optimization and selection and optimization together with a company in Austria. And then finally, the other idea we have is to inhibit the activation of the nerve ending, both at the periphery and in the CNS. And now I focus on these two channels, transient receptor potential vanilloid 1 and transient receptor potential ankyrin 1, which are related, relatives. And they are the capsaicin receptors, so we sense the pungency of the pepper by this TRPV1 and the pungency of cinnamon with the TRPA1. And if we block these receptors either directly or indirectly, we can inhibit pain. Okay? And then last year's medical Nobel Prize went to David Julius for the discovery of the heat sensing and mechanosensing receptors. But heat sensing is basically, of course, important, but these receptors are indeed important in pain. So actually, he discovered the TRPV1 receptor, and this is, they cloned this receptor in 1997. And this work was on the basis of my ex-boss, my mentor, János Szolcsányi, who is the father of capsaicin research, not just in Hungary, but all over the world. And actually, he also mentioned our professor in his Nobel laureate speech, and also the Hungarians working on this area for a long time. So here you have this TRPV1, which is activated by not just capsaicin and heat, but also protons and other inflammatory mediators. And the very, very closely interacting receptor is TRPA1, which is activated by not just cinnamaldehyde, but a lot of other endogenously produced molecules, such as, for example, methylamine, formaldehyde, hydrogen peroxide, methyl glyoxal, and acrolein, which are small molecule irritants formed from the deamination of different primary amines with the help of an enzyme called amine oxidase, copper-containing 3, so AOC3. These are deaminases. And actually the idea is either to block these receptors directly or block the formation of all the activators of these receptors, which is an indirect blockade by blocking the enzyme which produces these irritants. Okay, and together with a chemist in Budapest, Professor Péter Mátyus, we patented a drug called SV1287. This is szerves vettan from Budapest. So this compound is a unique compound which actually blocks this enzyme, which forms the irritants which activate the TRPA1, okay, in the tissues. But it turned out that this also directly inhibits TRPA1 and TRPV1 receptors, also direct blockade. And also it is metabolized to a metabolite, which is oxaprozin, which is a known cyclooxygenase inhibitor already in use for a long time in the US and in other countries. So actually we proved that this molecule effectively inhibits chronic neuropathic pain of different origin. We published a lot of papers on that, and here I just show you two graphs in an arthritis model. This is a chronic arthritis model which has neuropathic component. And in this arthritis model, you can see the inhibitory effect. This is the red line here of the compound during a 21-day investigation period. This is the hyperalgesia here, and also the edema formation. But which is interesting that in the late stage, there is no edema in this model, but there is still pain, which reflects the clinics. When the patients are successfully treated by the anti-inflammatory drugs, they don't have swelling, but they still have the pain. Okay, so this is effective here, and it also reduced the nerve injury-induced pain in the neuropathy model. And then we started the drug development of this molecule in 2014, And this is what we had for the discovery. We proved the effect of this molecule during 3 years, from 14 to 16, in different rat and mouse models. And then we started the preclinical drug development in 2016. We had the synthesis and the chemical characterization of the molecule. We had the drug technology, because we have to administer this drug in an enteric solvent preparation, because it's acid sensitive, and we don't want it to be cleaved in the gastric cavity. Then we had some acute and chronic tolerability study, safety study, toxicology study. We determined the pharmacokinetic parameters, which means the absorption, tissue accumulation, brain penetration, metabolism. And then we are just right now here, we had the authority evaluation at the end of last year. And we just started, with the help of a company in March, the phase 1 clinical trials on healthy volunteers. When we determine the safety and the kinetics, we are just right here. And then next year, if we are lucky and this molecule turns to be safe, we can test this in humans. And I just wanted to highlight here that original drug development is a very long process. So even if you are lucky, it takes at least 10 years. And for a university researcher, it is very hard, because it's very expensive as well. So you have to have industrial partners. So now, finally, the take-home message of this last bit is that we now have a small molecule drug candidate, which proved to be effective in different pain models. We have it in enterosolvent capsule at the moment. It is free of side effects. And I highlighted here that it's also free of the hyperthermic side effect, because these TRPV1 antagonists were developed by many, many companies for many, many years, and they were very effective. But their main problem was that because they also sense the temperature, they increased the temperature, the body temperature, and induced fever-like problems. But this one didn't induce fever-like problems. And it has a quite good kinetics. It gets into the brain, which is actually good for its therapeutic effect. So finally, we have actually started the clinical phase 1, which has nothing to do with the effect of the drug. It's not in patients. It's just to prove the safety of the drug and the behavior in the body. And we keep fingers crossed for the next few years to have a successful drug candidate, and also partners for the future development. And I would like to thank the team. We are, as you see, a huge team. And I'm very grateful for my colleagues for their efforts to put into all these projects. And I thank you for your attention. Thank you.
Thank you, Zsuzsa. And don't leave the stage, because we will hand over you the medal and the diploma.